Morning, ladies and gentlemen. It was my pleasure to welcome you to join the following section. We have three talks in the following section, including neuroscience and biological mimicry and public health. And the following speaker I'm going to introduce is Dr. Yi-Chun. Dr. Yi-Chun is a very special guest from Duke NSU Medical School, Singapore. And she's graduated from Taiwan University Physiology, and she finished her PhD in medicine from bioscience of physiology. Yeah, if I'm making wrong, you can make a correction later. And she's going to give us a talk about gut, brain, and emotion. Let's welcome her.
Okay. Good morning, everyone. It's my honor to today to present my work at this Brownland Week 2017. So I just briefly introduce myself. I received my degree in a bachelor's degree and a master's degree in psychology department in National Taiwan University. And then I moved to Germany to pursue my PhD. So I did my PhD in Max Planck Institute of Psychiatry. So at that time, I did a lot of the animal model of the psychiatry disorders. So today I would like to share my recent work. It's about the interaction between your gut and the brain, and how the gut microbiota influence our brain function and the emotion. Okay, so years ago, if you ask people, do you think that there's any dialogue between gut and the brain, then people will think you are crazy. Yeah, but I think most of people you hear about this saying that trust your gut feeling. So actually, yeah. So actually, there's the connection between these two organs. These two organs, they have a real connection based on the recent evidence, and how these two organs interact with each other. So let's have a look at the news in 2001. So it happened in a Canadian town called Walkerton. So there was a flood and caused the town's water become contaminated with the bacteria. And from the longitudinal report, they found that 8 years later, so the infectious irritable bowel syndrome was still high, and this symptom is also comorbid with some psychiatric disorder like anxiety and depressions. So like the example tell us, okay, so some disease in your gut is related to your brain function deficit. Okay, and in the last about 5 to 10 years, so increasing evidence have shown that various variation in the composition of the gut microbiomes may be associated with a change in the normal functional of your nervous system. Okay, so how the gut and the brain they have an interaction. So this interaction rely on the gut microbiota. And what's gut microbiota? It's a complex of the microorganisms that live in your digestive tract. And the number of the microbiota in and out our body is about 10 times of 10 times of your cell body. So you think you are the human beings? Maybe you are not. You are a working bacterial colony. Okay, and gut biota, we have a lot of gut biota in our body, and they are your good friends because they can help you to digest food. protect against the pathogen and also produce nitrogen for the host. And also it's helpful your normal immune system function. And most importantly, is my focus. The focus in my talk is very important for your healthy brain development and function. Okay, so how do we know that the gut microbiota influence your brain function? There are several research approaches, including aiding or removing the gut microbiota. So like if you keep the animals totally free from the bacteria, so then we can use the germ-free study. And also you can use the treat animals with the antibiotics and to kill some bacteria in your gut. And also you can see the consequence of the of aiding good or bad germs in your body and see how it influences your brain. Okay, so in my study we use the germ-free mouse model. So how to make the animals totally germ-free? So because we were born in sterile, but we were get the bacteria colonized during the birth. So how to avoid this cloning of the bacteria? So the first generation of germ-free, we will do the C-section. So just take the pups from the mothers and then put them in the handrail in the aspected isolator. And the subsequent generation that the germ-free animals is inbred and the mother can give the birth naturally in the isolator. So keep them totally free from the bacteria. Okay, so what's then if the germ-free mice they are getting adult? What's wrong with them? So the first signal we found is that they have a enlarged cecum. Can you hear? So when we weight the cecums, we can find in the germ-free mice, the cecum is like 5 times. 2.5 times. Yeah, it's 5 times larger, heavier than the SPF control. So SPF is the control group with a normal microbiota, and the germ-free they don't have any microbiota. Okay, so germ-free mice exhibit the enlarged cecum as a consequence of their absence of the gut microbiome. Okay, so then after we check that they are germ-free mice, then second we want to know if there's any behavioral change in these mice. So but mice they cannot speak. So how do they are anxious? They are smart, they are stupid, or whatever. So we need some tests, so behavioral tests to test their phenotype. So we use the open field to measure their locomotor activity. So see if in the open area, they are moving or they don't like move. So then we found that the lack of the microbiota induced the hypolocomotion. So that you can see, you can see here the distance traveled. The germ-free mice they travel much significantly less than the SPF control. And this open field also can test give us some information if the animals they are anxious. Because if the animals they are anxious, they will prefer to stay in the corner than the center. Because in the center they have a more risk to face the threat in the environment. So if they prefer to stay in the corner, that means that they might be more anxious. So here we found that the germ-free mice, they show less time in the center time. So that means they really spend more time in the corner, and that may give us some hint that they have some change in their emotion. Okay, so here's from the open field, we can find that lack of the microbiota result in hypolocomotion and increased anxiety-related behavior. So then later we want to make sure if they are really anxious. So we use a well-established paradigm, which is called elevated neural mass. So this mass is the elevated, and it contains 2 open arm and 2 closed arm. And the animals, they have they have a drive to explore the novel environment, but they are also afraid of height. So with these so they will face in this test they will face the conflict between the driven of explore the new environment and also a fear of the height. So if the animals that are anxious, they will spend more time in the closed arm rather than stay in the open arm. So here we found that the germ-free mice, they should stay in the closed arm, and so that means that the increased levels of anxiety-related behavior was found in the germ-free mice. Later, we also want to confirm, it's really true or it just happened to this test. So we use another anxiety test to measure their anxiety level. So again, we can confirm that the higher levels of anxiety-related behavior was found in the germ-free mice. And anxiety is always linked to the fear response. So that later we use the fear conditioning to test the animals' fear memory and fear learning. So we associated a tone, a neutral tone, and a shock for 5 times. So the animals will receive this fear conditioning training. And the data show that during acquisition phase, the germ-free mice displayed higher freezing behavior compared to SPF control. Then 24 hours later, we put the animals back to the different context and with the cue, and to test there if they have a higher response to the cue, which was associated with the shock. And then we found, okay, the germ-free mice also show the higher freezing response compared to the SPF controls. So here we can conclude that the lack of the gut microbiota not only leads to the elevated levels of anxiety-related behavior, but also the higher fearful response. Okay, so if you grow the animals in a bubble, so the germ-free environment, you will get the animals. They showed hypolocomotion and increased anxiety-related behavior and exaggerated fear response. So next, we want to know what's the mechanism of this behavior. And then first we we measure their stress hormone. And then we found that the animals, that they show higher levels of corticosterone level. So which indicates that the high HPA axis appears to be hyperactive in the germ-free mice. So the they secrete more stress hormone. So with the list of behavioral phenotype and elevated stress hormone, we want to know that if there's any brain region which is regulate this kind of this phenotype in the germ-free mice. So we look for the BLA, basolateral amygdala, because basolateral amygdala plays an important role in modulating anxiety and the fear response. Okay, so first we try to understand the neurons in the BLA, if there any change in the germ-free mice. So we use the Golgi staining so you can see here. So you can by using Golgi staining, you can see the morphology of the neurons. And we found that in the amygdala, there's a hyperoperization and higher spine density in the germ-free mice. Second, we want to know if not only the morphology, but also the neural activity is changed in the germ-free mice. And here you can see we use the N staining to mark this, to identify the BLA region. And then we found that after shock, the neuronal activity in the BLA in germ-free mice is elevated. Okay, so here you can see the significant increase. Okay, so here, first we found in the BLA, there's an increase in neuronal activation. And the second one, we want to know by using electrophysiology to see if there's any networking difference in the germ-free mice. So we use electrophysiology, and then we found that the frequency of the spontaneous EPSC is increased in the BLA in the germ-free mice, which means that the elevated functional connection between the neurons in these mice. Next, we also want to know with the same current, if the neuron can have a higher excitability. So then we found with the same current, the BLA neurons also have the higher neuronal excitability. So here is the summary. So growing up in a bubble, the animals will show these phenotypes, and also they have the functional change in their BLA regions. So our hypothesis that the lack of the gut microbiota will induce the brain's activation change in the BLA neurons, and also induce the elevated levels of the corticosterone, and then later induce the higher anxiety related behavior in these mice. So with this model, we can potential therapeutic, we can target some potential therapeutic way. So like we can do the conventional lines of the germ-free mice and try to put the bacteria back to the germ-free mice and see if this phenomena can be rescued. Or also we can change the corticosterone level or inhibit the BLA neurons activity to try to block and try to rescue the anxiety-related behavior due to the lack of the gut microbiota. Okay, so the application of my current study is because in Taiwan the resilience of the antibiotics is very high. So can you imagine? So I just check on the website. So we have 23 million Taiwanese people, but we come from more than 10 billion NT dollars of antibiotics per year. So it's quite a lot. So overuse of the antibiotics would induce the drug resistance. And then we would in our body maybe there was like super bugs that any antibiotics cannot treat. And also from my study, we can know that lack of some bacteria will induce the brain function and the mental health deficit. So I hope that my research finding can apply in the human clinical study and further help to the patients suffering mental disorder due to the imbalance of the gut microbiota, and also can maybe establish some future therapeutic ways, for these patients. Okay, so I would like to acknowledge some my current supervisor and the lab members, and also our cooperators, and they helped in breeding the germ-free mice. And yeah, so thank you for your attention, and yeah, I'm ready to any question. Thank you.
Thanks, Dr. Yen, for interesting research. Now we know the relationship of bacteria in our guts and our mental condition. Is there any questions? Yeah.
Okay, so we've already know that the distinctions between the GF mouses and the standard. Oh, GF. I forgot.
It's okay. It's okay. GF.
So I am Yi-Chun Yen from Tzu Junior High School,
Hi.
And senior. Anyway, don't really matters. So, do we know which kind of bacteria will create certain behaviors?
Bacteria will
create a certain behavior.
In my study, that we clear out all of the bacteria, and we found that the increased anxiety-related behavior. But there are some bad bacteria will induce also anxiety phenotype. Yeah, they have some that infectious study, and they just get infectious with some bacteria, and they found that as the similar anxiety phenotype. But maybe the kind of anxiety and this kind of anxiety, they have a different underlying mechanism. Yeah. So, but now I'm focusing on the germ-free anxiety phenotype and try to understand.
Yeah, it's kind of, you have lots of good bacteria and bad bacteria in your stomach. So that's the next question I want to ask. If you want to put some bacteria back to germ-free mice, what kind of bacteria you will put back?
Here we just conventional. I can show the next one. Actually, okay. Okay, so how we do the conventionalize. So you can see here. Here's the parent. They are conventionalized, right? And then we just put a co-house the germ-free mice with SPF mice. The SPF they have a normal gut bacteria and co-house for 2 weeks, and then let them stabilize their colony of the bacteria, and then when the germ-free mice, they are normal germ-free, then we just cross over and have everyone conventionalized offspring.
Yeah.
Yeah. So supposedly, so the conventionalized offspring have all kinds of the normal that bacteria SPF have. Yeah.
Any other questions?
Yeah.
Yes.
Very interesting research. The question I have about is it only the direct effect between the gut and the brain?
Mm-hmm.
Because we know the gut is responsible for producing a lot of vitamins and stuff, and maybe lacking these vitamins could have an effect on increasing anxiety and stuff like this. So if you have a reduction in certain vitamins, people will have different symptoms from healthy population.
Yeah.
So have you looked into other parameters to see if they suffer from deficiency in certain proteins or certain enzymes? Or
actually, we haven't checked that far. We just right now. I think is like a model that how the gut so influence the brain. Yeah, okay. And there's some pathway. And right now we focus on the stress hormone system. Yeah, but of course maybe because I've done one experiment. I used the chronic treatment of the corticosteroid inhibitor. Yeah, but I can't. I can reduce the corticosteroid level, but I can't I can't reduce the anxiety level. Okay. So, which means maybe the anxiety is due to not only one factors. Maybe other factors also involved in this mechanism.
Okay. Any more questions? If no, then we thank Dr. Yang. Dr. Yen, okay, thank you.